Assalamu alaikum. My name is Zain from the Department of Biochemistry. Today I am going to present my presentation of Principle of Chemistry 1, whose topic is L-isoleucine. These are the table of contents of my presentation. First of all, I will tell you what is L-isoleucine, introduction of L-isoleucine. L-isoleucine is an isomer of L-leucine and is essential amino acid. It is synthesized from thurine and is branched chain hydrophobic amino acid. L-leucine is basically an isomer of L-leucine, which is an amino acid, and amino acids are building blocks of protein. Hence, it is present in proteins as essential branch chain aliphatic amino acid found in many proteins. It is isomer of leucine. It is important in hemoglobin synthesis and regulation of blood sugar and energy levels. As we know, what is hemoglobin? Hemoglobin is a oxygen carrying pigment on red blood cells. L-isoleucine has important role in the synthesis of hemoglobin on red blood cells, and it also have a role in regulation of blood sugar level. Next is the structure of isoleucine. What is L-isoleucine structure? Isoleucine is an alpha amino acid that is used in the biosynthesis of protein. It contains an alpha amino group. Here is the term alpha amino group. Alpha amino group means the amino group. This is amino group which is attached to the alpha carbon, and alpha carbon is that carbon which is directly attached with the functional groups and a organic compound. With the carbon is directly attached to the two functional group, which is one of them is amino acid and two other is a carboxylic acid. The other is central carbon, which is attached with a carbon and a hydrocarbon chain. Here is a hydrocarbon chain, and this is a second carbon atom. This is a zigzag structure uh, of the L-isoleucine, an alpha carboxylic acid group and hydrocarbon side chain with branched central carbon atom bound to three carbon atoms. This is also a three-dimensional model structure of isoleucine. The next is the preparation method, how we can prepare isoleucine. First of all, it is classified as a nonpolar. Like other organic compounds, it is also a nonpolar. Uncharged mean it is a neutral compound at physiological pH. Branched chain, it is branched chain as we discussed in early slides. Aliphatic amino acid, it is essential in human, meaning the body cannot synthesize. Our human body is unable to synthesize isoleucine naturally in it, so it must be ingested in our diet. Isoleucine is synthesized from pyruvate, implying isoleucine enzyme, biosynthesis enzyme in other organisms such as bacteria. Like insulin, we prepare isoleucine in bacteria. It is encoded by the codons. These are three codons which are responsible for the formation of isoleucine in bacteria. These are adenine, uracil, adenine, uracil, cytosine, adenine, uracil, and adenine. The reaction starts with the pyruvate molecule. This is a pyruvate molecule. Applying the enzyme, leucine biosynthesis enzyme, it is converted into that organic compound. This organic compound is then converted into leucine. On the isomerization of leucine, it is converted into isoleucine, and this is the final product of this reaction. Next are some properties of isoleucine. The first property of isoleucine is biosynthesis. Biosynthesis means the synthesis of this amino acid in living organism. As an essential nutrient, it is not synthesized in body, hence it must be ingested usually as a component of protein. As we discussed in previous slide, it is not synthesized in our body. So that's why we have to ingest in this amino acid with other protein diets with our food. In plants, microorganisms, it is synthesized via several steps, starting from pyruvate, as we see in the previous slide. Reaction start from pyruvate and then converted into alpha ketobutyrate and then into leucine, and on isomerization of leucine it is converted into isoleucine. Enzyme involved in this biosynthesis include acetolactate synthase, also known as acetohydroxy acid synthase, acetohydroxy acid isomer reductase, dihydroxy acid dehydratase, valine aminotransferase. The next property is catabolism of L-leucine. L-isoleucine is both a glucogenic and ketogenic amino acid. These are two types of amino acid, glucogenic and ketogenic. After transmission with alpha ketoglutarate, the carbon skeleton is oxidized and split into propionyl CoA and acetyl CoA. The carbon skeleton of isoleucine in the catabolism is converted into two compounds called acetyl-CoA and propanyl-CoA, in which one of them, propanyl-CoA, is converted into succinyl-CoA, a TCA cycle intermediate which can be converted into oxalate for glucogenesis. The succinyl-CoA is an intermediate of TCA cycle. The TCA cycle is a cycle in which isoleucine is produced. Next is the property insulin resistant. Like other amino acids, isoleucine is also insulin resistant. High levels of isoleucine are observed in the blood of diabetic mice, rats, and humans. This shows that the humans or other organisms which are suffering with the diabetes have high level of isoleucine in their blood. Mice fed and isoleucine. This is an experiment conducted on diabetic mice uh, to observe the role of isoleucine in living organisms. Mice fed an isoleucine deprivation diet for 1 day and have improved insulin sensitivity, and feeding of isoleucine deprivation diet for 1 week significantly decreased the blood glucose level. In diet induced obese, the insulin resistant mice, a diet with decreased level of isoleucine with or without other branch in amino acid result in reduced adiposity and improved insulin sensitivity. Next is the IPAC name of this compound. The IPAC name of this compound is isoleucine, which is the topic of my presentation. Other name of this compound is 2-amino 3-methyl pentanoic acid. Formula, molecular formula of this compound is C6H13NO2. The molar mass of this compound is 131.17 grams per mole. Next are the uses or application of this amino acid. Athletic performance. One study found that the mixture of amino acid, which include isoleucine, improved the training efficiency in players. We often see the players or gym workers take protein shakes, which are made up of protein powder. This protein powder include isoleucine. Research found positive change in muscles integrity and hematopoiesis, the formation of blood cells. In these athletes, the study concluded that amino acids not only support muscular fatigue recovery but also increase the blood oxygen carrying capacity. As we discussed in the first slide, that it is responsible for making or synthesis of oxygen carrying pigment, hemoglobin, and red blood cells. So it increases the number of hemoglobin pigments on red blood cells that carry oxygen. So that's why it increases the oxygen carrying capacity of muscles in athletes. This likely helped these athletes improve their performance. The next use is to regulate blood sugar. Isoleucine is essential for blood sugar regulation, and isoleucine deficiency can mirror the effect of hypoglycemia, which means the low blood sugar level. The third use of this is protein synthesis, promote an anabolic response in muscles and decrease the rate of protein degradation. Research have found that BCAAS stand for branch chain amino acids consumed before and after exercise initiate the adjustment to protein synthesis by making changes to signaling pathway involved in the protein synthesis. They have also found that the leucine produced a better response in protein synthesis than its BCAA counterparts, but taking leucine alone is not enough. Therefore, bodies suggest that taking all three BCAS branch chain amino acids together achieve desired protein results. As we know, everything which has benefits also has the side effects. Here are some side effects of isoleucine. Very high doses of leucine may cause low blood sugar, hypoglycemia. It may also cause Pelgra disease. The symptom of this disease includes skin lesion. As we see in the first picture, this is the skin lesion disease, which is symptom of Pelgra disease. It also causes hair loss, as we see in the second picture, patches of hair loss in head region, and gastrointestinal problems like constipation, diarrhea, indigestion, and stomach pain are also symptom of Pelgra disease. At last, it is a conclusion of my presentation. Isoleucine is essential amino acid that is not synthesized in our body and have to take our with our diet. L-isoleucine with other three amino acids of BCAS branch chain amino acid is very important for athletes and gym workers. Deficiency of isoleucine result in several disorders like muscle fatigue, etc. Thank you. That's all from myself. Thanks for your attention.